I'm going to be talking to you this morning about thinking about how we sort of apply and interpret polygenic scores, and in particular how we think about this at scale. So not just in kind of small limited settings, but how we think about this across a health system or across a sort of national biobank kind of scale. And a huge thanks to the organizers for inviting me. It's a real honor. My name is Gignoux. I'm at the University of Colorado at the Anschutz Medical Campus, where we're interested in a lot of kind of these similar methodologies and working in large research networks and that sort of thing to gain new insights in personalized medicine. And I wanted to talk a little bit about sort of our interest in personalized medicine and how this kind of factors in. And just to say, the center that a lot of us are embedded in at the University of Colorado is sort of a joint venture of our university and our health system together. So we've got multiple sets of bosses, but it's important to kind of think about this in the context of like, well, so when we're thinking about risk prediction and that sort of thing, how does this both affect opportunities for research and potentially getting into clinical implementation? So of course, kind of a hotbed topic these days is around what personalized medicine is. And it's this idea that, we can sort of tailor kind of treatments and regimens and that sort of thing to each individual, and hopefully, provide sort of better access, lower cost, better health outcomes for individuals by using this kind of rich data that's able to be generated at scale. And so, we've been thinking about this in different contexts, of course. There's certainly a lot of interest in the kind of clinical side into that, high-impact pathogenic variants, some of the work that Emer Kenny was talking about yesterday as an example. And then the stuff that I'm going to talk about today a little bit more kind of focuses on genome-wide association study insights. And so a lot of people are sort of thinking about this in the context of research, and then, how do we translate these findings? And one of the things that's, worth knowing is, for those of us who were in the field when this stuff was starting to come together, this started as a fairly rare thing across the entire genome. Of course, as time goes on, we identify more and more genetic signals, and of course now we've got thousands, tens of thousands, hundreds of thousands of associated variants across a number of disease domains. And so the interesting thing then is to think about how we take that information, all of these insights, and kind of distill them in a way that is relevant for, say, a single patient walking into a clinic. But there's a lot of, kind of critiques of GWAS, particularly in talking about, in talking with disease domain experts and people sort of, working at the bench and that sort of thing. So, of course, we know that there's a lot of weak effects that are found, right? It's rare that you find an odds ratio above 2 or even above 1.5 for most complex traits. Maybe it finds uninteresting genes, things that sort of potentially act in sort of unintuitive ways or ways that are difficult to ascribe to a specific molecular pathway. And then, of course, there's these questions of like, how much of the genome is actually associated with different traits. There's been debates on that. And of course, there's a lot of kind of concern that a lot of this work is kind of done, with extremely broad phenotypes. So only case control when everyone knows that there's disease-specific heterogeneity in a lot of complex diseases that oftentimes people aren't taking into account. So there's a lot of examples of that kind of sledgehammer-based approach that makes things kind of difficult to gain insight. And of course, these are expensive, right? But this kind of work these kind of assumptions kind of rely on treating every variant kind of on its own, which is oftentimes what people do in a genome-wide association study. But that's not really the case when we think about the sort of aggregate genetic effect across the entire genome. And at that point, you get to a situation where the genetic effects in aggregate, sort of the overall genomic picture of this kind of information, isn't weak. And so, when we think about how this affects kind of the marginal testing in genome-wide association studies, they find weak effects. Okay, well, in aggregate, those weak effects turn out to be maybe one strong predictor of risk. The question of which genes and how many genes and that sort of thing becomes less of an issue. And then we can think about specific context, right? And I'll talk a little bit about sort of context in a second as well. But now we're no longer, because there's a lot more statistical power, we're looking at a stronger aggregate effect across the genome. Maybe there's a specific setting in which we can think about a particular sort of narrower phenotype definition, or something that's sort of more clinically relevant, say, than a case control study, thinking about, say, tumor progression in the case of cancer, or other kind of sort of subphenotyping that may have more immediate clinical impact. And again, because of the statistical power that these kind of the sort of thinking about the genome all at once allows us to do, we don't necessarily have to think about expensive generating data in hundreds of thousands of people. Now we can think about a setting where potentially there's a couple hundred individuals. It sort of changes the game in terms of this kind of budget. So this is probably not too surprising information for a lot of people in this audience, but just so we think about this, oftentimes when people are interested in polygenic scoring, as it's called, people are thinking about sort of a linear way of distilling this information. So you have to do something to deal with the linkage disequilibrium that's present on all the chromosomes. And there's different kind of algorithmic ways, some that only choose a specific SNP, or some that attempt to marginalize out the effects of LD. And then you take these effects across the genome with a certain weight for each site that's considered in your model, and that gives you a score. So it's a very simple and straightforward kind of approach. And a lot of what we think about is the kind of predictive ability that we have to derive these scores and actually use them. This is now from a paper that's about 10 years ago, but it sort of goes along with kind of what we expect in terms of prediction in general in sort of biomedicine. But it's the idea, to have a sort of aptly powered study in discovery to sort of precisely capture the effects. You want a disease that's certainly, or a trait that's fairly heritable. If something's unlikely to be heritable, then it's unlikely that this kind of polygenic scoring is going to give you much predictive accuracy. So cool. I mean, we're done, right? Like that's, there's a lot of GWASs, we have a lot of scores, and so we can just apply them. We can generate these genome-wide array or sequencing data sets. We can just apply a bunch of scores and we're done. But of course, it's not that simple. Or really this talk would be like 10 minutes and it would be like, here's a couple of Plink commands and you're done. And so we have to sort of think about what this kind of information means. how to use it, and especially how to use it in sort of responsible kind of ways. And so that's where this idea of context becomes really important when we start to think about how to interpret these, and in particular how we think about the opportunities for clinical implementation. So this is now from a review, again, about from 10 years ago, and sort of describes why we see so much interest in polygenic scoring now versus, say, like 5 years ago. And it's because there's this nonlinear relationship between the sample size here on the x-axis and how close your R-squared can approach heritability. And so it's, because of this nonlinear thing, we see sort of this, sort of sigmoid function. So, we may not be necessarily at like approaching heritability for a lot of traits, but we're now getting to a point where there's large enough sample sizes in discovery to kind of think about this kind of information. A really nice paper now from a couple of years ago kind of describes, in addition, how a lot of this information is not sort of static. How we think about applying these and their impacts on things like heritability and the polygenic scoring, sort of R-squared or prediction accuracy, how that information changes over time, how much it changes dependent on the data that you include has major implications for what this data actually means, and particularly what it means in a prediction context. So all of this stuff becomes important. I'm going to talk a little bit about sort of global pictures of this same kind of context. There's been a huge amount of interest in genome-wide association studies. Now it's to the order of like 1 in a couple hundred people have participated in some sort of GWAS across the world, which is a very high number. And there's been a huge amount of interest in this work, both from scientists and from the lay audience across a number of different sort of trait domains, and sort of thinking about kind of broader implications in society. But one of the things that's important to note is at the same time, there's a lot of interest in kind of, not just within polygenic scoring, but in general, kind of thinking about how people in general think about prediction. All of this kind of fits into a broader scheme that goes along with a lot of kind of the insights that have happened in machine learning and kind of other aspects of prediction. And it's important to note that all of these kind of ways of thinking about prediction accuracy and context and everything sort of falls into a broader kind of category of current research. And there's a lot of really nice papers now kind of thinking about kind of developing pipelines, how we think about kind of running these kind of scoring regimes and sort of ways of thinking about doing it, ways of ensuring that your data is high enough quality to get accurate polygenic scores. I'm just providing some examples of these kind of studies here, and happy to share slides with folks who are interested. So here's an example now from a widely cited paper from about 4 years ago, kind of showing this is actual real data from UK Biobank. It's not, even though it looks like a sort of very idealized distribution, it sort of comes from the fact that this is a large number of individuals underlying this distribution. And so within that, then, you can find people that are at very increased risk of developing coronary artery disease, right? So, depending on where you set your cut point, you can identify a subgroup that has twice the odds, 3 times the odds, however far you want to get into the extreme, you can get, 10 times the odds. And so, as that information, as those patterns of risk become higher and higher, they become more and more, sort of broad interest. People are paying attention to them. And it gives some really exciting opportunities, both in terms of, the kind of initial findings and categorizing this risk, and also thinking about it in terms of sort of pragmatic trials and other opportunities to use this information downstream. And maybe that helps you identify individuals that could benefit from additional therapies. So here's one of these examples. So sort of moving beyond just predicting risk, asking whether there's certain therapies that a subgroup will benefit from, in this case kind of informed by polygenic scores. So a lot of this information is here, and it's available to a lot of people. So there's a huge caveat, of course, with this. So a big but in red there. And that's when we start to think about who is taking part in these genome-wide association studies. And it's probably not a surprise to a lot of people in the audience here, sort of people who have been thinking about human genetics for a long time, that most of the information that we have is from a single continental group, right? It's of individuals of European ancestry. And so certainly, there's a disconnect in terms of what we know across different populations, and there's sort of relative representation in terms of populations across the world. And this has major implications for how we think about this polygenic scoring kind of regime, or how these pipelines come together, because oftentimes necessarily you're sort of focusing on the red group here, and that doesn't necessarily represent all populations around the world. And so, it's important to kind of keep this information in mind. From human origins in Africa, our species has had an incredibly complex history. And of course, this is only sort of one slice of this expansion around the world. Human genetic history is incredibly complex, right? There's a huge amount of migrations, and particularly if we think about sort of movements of people, both voluntary and forced over the past 500 years, it's really sort of changed the genomic landscape around the world. But the important thing to note here is that all of these kind of demographic effects play out in the entire genome. So this is why when people run genome-wide association studies and they correct for ancestry, or correct for ancestry using PCA, like the first couple of PCs don't tell you about case control status or that sort of thing. In most instances, they tell you about ancestry, and it's because all of these migrations and movements of individuals shape, even through neutral processes, the entire genome. And so we've been interested in this work for quite a while. And so one of the things that we were interested in doing is just kind of understanding early on kind of implications of these polygenic scores. This is now from a paper from 5 years ago, where we kind of tested the existing polygenic scores at the time, even for relatively sort of well-characterized traits that are highly heritable, such as height. And when you do this, this is just looking at 1,000 genomes populations, so these are just the distributions of the polygenic scores. What you see is that the sort of inherent population structure across groups leads to these differences in distributions across different populations. And so, within each distribution, you still expect there to be some degree of prediction accuracy, but that means that you have to know the individual and the right group to compare them against, right? Again, this is sort of speaking to this broader question about why context matters so much. And so, we can think about an individual here sort of on, as one sort of static line, but on its own, it doesn't necessarily matter without kind of understanding the relevant ancestral group to compare against. So we can think about people here who have very different sort of absolute values to their scores, but maybe they're still within sort of the middle of the distribution for their population of interest. Yep.
Can you explain this plot again? What we're seeing here exactly?
Yeah, so this is just taking a height polygenic score based out of giant and just asking what do the distributions look like of those scores in 1,000 genomes data.
And what are the polygenic score units here? Just height relative to some average, or?
Yeah, it's a great question. So sometimes these are calibrated to centimeters or something like that, and oftentimes they're not. So oftentimes we sort of think about polygenic scores on their own as being kind of unitless. And maybe, because part of it depends on how people reweight the betas that go into the models and that sort of thing. So yeah, I mean, in this kind of context, this would be sort of a more abstract score, and you'd have to know something about the, sort of mean and variance of each population.
Do you expect it to correlate with height? So, like, if
Yeah.
I were to run this blindly, then I would probably be underestimating height of, say, people with African ancestry.
Right. Well, that's the challenge. If you don't take this ancestry information into account, then yeah, maybe you think that like African and East Asian individuals would be predicted to be like 3 feet tall or something like that. And obviously that's not the case. But that's why it's important to think about what's going on for each individual in each sort of population distribution in this kind of context.
But does per distribution, are they like ranked, sorted reasonably, or does that even get screwed up?
Yeah, I'll talk about that a little bit more in a second. But yeah, so if you know the individual and you've got the right sort of population kind of distribution to compare against, then yes, you expect that these scores are still going to have some degree of correlation to the outcome within groups. But like the sort of within group comparison gets swamped by the degree of population structure that gets baked in if people aren't careful. So it's sort of critical to kind of think about an individual's score in this kind of instance, and not just their score on their own, but kind of where people fall into these kind of quantiles, right? This is sort of a toy version of what we saw before in real data. But all of this is to say that all of this information affects what we can do and how we can interpret these kind of findings in sort of a precision or personalized medicine context. So this is going back to your question from before. This is some really nice work from Alicia Martin and colleagues kind of demonstrating that not only do we have these sort of have this ancestry stratification effect, but the fact that a lot of this work has been done only in populations of European descent leads to a situation where the sort of prediction accuracy kind of falls off as a factor of the genetic distance or the degree of genetic similarity between certain populations and populations of European descent. And this is across a number of different traits, across different disease domains, and this work has been now widely replicated by a lot of different groups. So without kind of thinking about polygenic scores in the context of genetic ancestry or other kinds of ancestry measures, these scores are going to be highly confounded, and you're going to be stuck in this situation where your prediction accuracy tends to fall off in populations that are more different from the population where the discovery took place. And since a lot of this work is largely in populations of European ancestry, there's a major concern about perpetrating health disparities by just sort of naively applying these scores in sort of a personalized medicine or clinical context. So in the last part of my talk, I'm going to talk a bit about some of the strategies that we've been thinking about and some of the sort of ongoing work that we've been doing to kind of mitigate this information or these challenges. And so, there's a couple of different ways of thinking about this. One of the things, of course, is like, well, let's try to get a lot of data together. Maybe we can sort of counteract some of these effects by bringing together a large number of groups and sort of focusing on populations that aren't of European descent. This is something that we've been involved in for a while. As part of the PAGE study, I was talking to some folks about this work this morning. This originally started as a sort of genome-wide association study and replication consortium involving a large number of different populations. But of course, sort of thinking about this information categorically is not necessarily sort of reflecting the underlying spectrum of ancestry that exists in these individuals. So even to the point where, in this case, looking at principal components 7 and 8, we can kind of disentangle sort of fine-scale ancestry across the Americas. So kind of taking all this information into account requires really large data sets. It involves some of our buddies at UCLA, where we've been trying to put together a large consortium involving studies across the world in multiple biobanks, and even partnerships with direct-to-consumer companies to try to put together data sets in the hundreds of thousands or millions of individuals for this kind of work. And it's been really exciting to see people kind of leverage this data set and gain new insights. And of course, in PAGE, we're not the only ones doing this. It's important to note now NHGRI has put a major commitment towards better understanding this kind of information through the PRIMED consortium. And again, this is UCLA is one of the PRIMED sites. So there's a lot of interest in kind of building these larger resources and kind of getting around this information gap in this kind of context. That's only kind of one way of thinking about this information. One of the other things that we can do is we can think about simulations or other aspects of mathematical modeling that allow us to kind of, generate, hopefully realistic data in these kind of situations and test different association study methodologies, for example. And so one of the things that we've been interested in this kind of context is situations where there's ancestry associations with quantitative phenotypes, and this happens quite a bit. This is an example from baseline skin pigmentation from a paper that we published a few years ago. And one of the things that we're sort of interested in understanding is there's a good degree of population structure, so as measured by FST, at different trait-associated variants across the genome. So here's an example of almost 900 traits from the GWAS catalog, and the FST in this kind of instance between a West African population and Europeans from 1,000 genomes, and kind of describing the fact that sort of background FST is this dashed line across all these traits. And so we see a lot of trait-associated variation that is more differentiated than background. And so it's important to kind of think about like, in general, maybe a lot of these variants have relatively low FST, but every now and then you're going to find some that are highly differentiated. And so we worked in collaboration with Brenna Hen's group and Noah Zaitlin's group to put together a simulation framework, and this is all work that's led by my wonderful postdoc, to generate a kind of mechanistic simulation that allows us to kind of model these ancestry phenotype associations and the degree to which they're driven by genetics versus environment. And one of the things that we show under this simulation framework is that, you know, people have thought in general like, oh, working in populations of mixed ancestry, working in structured populations is hard, and so people have avoided it. It's part of the reason there's been such a Eurocentric bias. But in general, populations that do have some degree of mixed ancestry are expected to have greater statistical power for the same size GWAS. And so here's comparing in red the population of mixed ancestry to the two parental populations in green and blue, and you can see the power gains that are possible both in quantitative and dichotomous traits. So this has implications then for how we think about sort of, again, turning these insights into polygenic scores. So as we can see in this kind of example, then polygenic scores that are derived from the admixed population tend to do better in different groups than scores that are only trained on a single population. Yeah? Oh, yeah. So it's because in populations of mixed ancestry, and you know, particularly sort of the more admixture that exists, the more it's likely that these variants that are highly differentiated are going to be at sort of intermediate allele frequencies, and so you're going to have maximal power for those specific variants, and sort of not much of a power loss necessarily for the rest of the variants. And so that's what's really driving this, that boost in power.
Another question regarding the simulation framework.
Mm-hmm.
Are you trying to model kind of differential selection, and trying to model kind of different environments that might lead to like the large FSPs?
Yeah. Yeah, it involves sort of thinking about the relationship between, sort of ancestry-associated selection and how that impacts different populations differently.
So the simulations try to model that, have
Mm-hmm.
different regions under selection, maybe?
Yeah. It's a relatively simple simulation in that it treats the sites independently, so there aren't necessarily regions. It's just thinking about causal variance and non-causal variance for a specific trait. But then it allows you to sort of change the dial from something that's ancestry associated and due to environment versus ancestry associated and due to genetics, or anywhere in between. Just wanted to finish up in the time I've got left and talk a little bit about active research that we have going on now, kind of thinking about how do we apply these scoring methods, and what does this tell us about polygenic scores based on what people have already gathered. This is really helpful in this kind of context that there's been a lot of work sort of putting together a catalog of polygenic scores. This is kind of analogous to the GWAS catalog. So you can go onto pgscatalog.org and rapidly download a bunch of scores, how they were devised, aspects of like the number of individuals that went into discovery and that sort of thing. So it's a really, really fantastic resource. And then we sort of benefited at the University of Colorado because we're a new biobank, and we haven't participated in any of these specific sort of traits and polygenic scores yet, although now we're definitely part of it. But so we were sort of able to take this as a snapshot in time when we were brand new, so we're out of sample. We don't have to worry about overfitting. And we're a data set that includes representation from multiple ancestry groups, and so we can think about how this information actually plays out in terms of the different populations that we have in the CCPM biobank. So again, kind of translating this data into something that we can kind of use systematically is not necessarily simple. We want to be able to kind of harmonize this data across a number of, genome builds, whether people are on the right strand, all of that kind of stuff needs to be harmonized. We have a sort of working version of this kind of information now. And so then, that gives us an opportunity to look at hundreds of scores across thousands of different phi codes. So we're able to kind of, do the kind of all traits by all scores kind of comparison. And so it becomes an extremely rich data set. One of the things that we can, do then is sort of, this is a meta-analysis across all the different populations, but this is sort of a, this looks like a standard PheWAS plot, but what it describes is the sort of most significant trait by score relationship for any specific point. So we can identify the scores that kind of fit are most strongly associated with each phi code. But this is, so this is kind of a 3D PheWAS plot almost. But one of the things that we can do then is we can start to think about what scores are most heterogeneous across groups and which are most homogeneous across groups. And so we're measuring this using the I-squared heterogeneity statistic. And we can see that there's a huge distribution of scores, right? Some that are extremely heterogeneous and some that have an I-squared approaching 0. So there are some scores in the PGS catalog that don't necessarily have the same sort of issue of information loss. One of the things that's worth kind of highlighting, though, is it depends on how you measure this information. And this is kind of consistent with work that came out of the Pasanyuk lab, kind of describing that there's a good degree of uncertainty in polygenic scores that people don't think about when they only look at the point estimates. What we see here is that there's a relationship between how you, whether you parameterize your sort of measurements on per standard deviation of the score and whether you only think about sort of outliers. If you're interested in, say, the top 5% or the top 10% of the distribution, that you're not necessarily getting the same information. And sure, there's some correlation, but it's important to note that it's, it certainly isn't necessarily explaining this. And this becomes important when we think about personalized medicine because maybe there's individuals that you want to, yeah.
Could you kind of, I'm not sure how to think first question, what does that mean? How is that calculated?
It's the, yeah. Yeah, and it's measuring the sort of degree, like the sort of beta of the association between, for a certain population group and the trait of interest. Yeah, so it's the random effects meta-analysis kind of measure of heterogeneity.
Heterogeneity across populations.
Across populations, exactly. Yeah, thanks. That's important to clarify. But then, how we think about this changes, right? And you can see kind of along these axes that there's a good amount of scores that appear to be highly heterogeneous dependent on one measure of how we think about this stuff and are completely homogeneous on the other axis. But just to give some examples of this as we start to kind of think about this, so there's certainly instances where the I-squared is very high and, we're sort of looking at situations here. We've got Europeans in CCPM at the top, where there's a good degree of heterogeneity. And in fact, in some populations, your point estimate would sort of go in the opposite direction of what you would expect. That's not the only example. We have a large number of examples like this in the paper that we're writing up right now. There are some instances where there's still a high degree of heterogeneity, but all populations kind of are, predicted sort of aligned on the same axis of risk. But the other thing that's kind of important to note is that there's situations where you see kind of fairly homogeneous scores. And certainly these are the exception, not the rule, but having access to this large catalog of polygenic scores allows us to identify specific situations like this. So here's an example of the relationship between sleep apnea and body mass index. And here is a score for breast cancer, where things are looking pretty promising, and a score for type 2 diabetes here. And so one of the things that we're actively working on now is kind of characterizing these patterns and what is it about these specific scores that make them different from these more heterogeneous kind of scores. And so with that, polygenic scores are a very exciting kind of new development, or not even new at this point, but kind of, a lot of people are thinking about moving them closer and closer to the clinic, so there's a lot more interest in a lot of different contexts for these. So there's a lot of opportunities just beyond kind of disease discovery, kind of thinking about therapies and other kind of context ways of studying this information. And it's really nice to have access to biobank data for this kind of thing, because you can look across the entire phenome. But in terms of the kind of applications, there's a lot of challenges of how do we understand ancestry, how do we model ancestry, and how do we use that information appropriately, and then try to find aspects of scoring regimes that allow us to have less heterogeneity in our measurements. And of course, that leads to this final thing where, anytime that we think about how this has implications with patients in the real world, with anyone walking into a clinic, it's not just as simple as like having a score and running a dot product and you're done, right? All this context stuff is extremely important. So with that, thanks very much. Just wanted to highlight the work from my lab and collaborators across Page that have helped with kind of thinking about this stuff. And thanks everyone for being here.